performing an analysis of mutation calling viral genome reconstruction and lineage clade assignment from SARS-CoV-2 sequencing data by Genomics 2C group. Hello, I am Nuiter, and this analysis is the third week task of the Hakbio Internship. In this case, our group, Genomics 2C, has chosen the tutorial prepared for Galaxy project by Wolfgang, Maier, and Bérénice Batut, corresponding to a current topic such as the identification of the different emerging variants of SARS-CoV-2. For this tutorial, a set of SARS-CoV-2 samples hosted in the Zenodo repository and a set of auxiliary datasets have been selected to carry out the analysis, as we can see in this graph. 3 workflows have also been used corresponding to the different steps carried out on the Galaxy platform EU server, and 2 more tools for the final analysis. To start, we will create a new history in Galaxy, rename it, and upload the files needed to perform the analysis, samples, dataset, and auxiliary datasets. Galaxy, we have a new history created. Name it. Now we must to upload the necessary data. Samples, and we must to select the appropriate files type. .pc, fastQC, and your fastGC, and now the auxiliary data. We must to wait for a few minutes. Now we will create a pirate collection from samples datasets. Selecting all and unselecting auxiliary files, we must to build a list of datasets pairs. We must to pair all the appropriate filter for forward and reverse, and a name for the list. Perfect. Here is it. And now we must to be sure that the format of the auxiliary datasets are correct. TSV must tabular and is txt, and we need to fix it. In data types, looking for tabular, select tabular and change data type. Now, this is interval and must be bed. Same way. Bed and change. TSV is tabular, right? And reference FASTA is FASTA, right? Okay, perfect. Now we must to load the workflow. In workflow menu, import search form. Tier is server doc store and search for organization EWC workflows and choosing Arctic P Illumina variant column. This is the appropriate and the last version. Perfect. And now we can run the workflow. The pirate collection is the input dataset, FASTA sequence, the reference is reference FASTA, the bed file, primers, and the amplicon assignment. The other option must be remain like default. And now run the workflow. This workflow take around 2 hours, so we have to be patient. But fortunately, I have finished this step in another history. But for now, we can see how is the process. Okay, the workflow creates a lot of uh, files, result of different process, and this workflow is scheduled around 28 steps. It's working properly. And now we go to change to the another history with the process finished. Okay, here we have the process finished with all the files created for the workflow. And now we must apply another one workflow following the mechanics see above. Import search form text store and the query. And now variation reporting. Selecting the appropriate version and run it now. Okay, here we must to change for annotated variants, and here we must to change to featuring mapping and number of cluster 3 and run workflow. This task take around 1 hour and we must wait again. Okay, the second workflow has finished. And now, once we have finished the previous step, the next step is from ABs to consensus sequence, for which we will follow the previous instruction to import the corresponding workflow. Workflow, import, search form, query, text store, and now we must choose consensus construction. And run it. For variant calls, we choose final, it's okay. Fully process rate, 5, and it's okay. Okay, and now run workflow again. This is the third and the last workflow. We must wait for it. Now, with these 3 steps, we finished the processing of our data. To now proceed to the analysis using Pangolin and Netscape tools. Just a minute. and perfect. Finish it. Okay. For pangolin, we can look for it in the search bar. So pangolin, and we must to left dollar latest from web or pangolin source, and using the multi-sample consensus FASTA. Execute. All right. Now pangolin is finished too, and the last one will be next clade. Looking for it. I'm checking for the correct. Right, and output option tabular format report. It's perfect. We must wait a little bit. And now it is finished. And with these steps, we finish the procedure in the Galaxy platform. The conclusions drawn will be provided in annexed documents. Thank you very much for watching this video, and I hope you enjoyed it as much as I did. Thank you. Bye bye.